While she's setting up, let me introduce myself. I'm Nitin Baliga. I'm a faculty member here, and I also serve as the Senior Vice President and Director of ISP. And I'll be speaking to you about resilience and drug tolerance in microbes. I think the first two speakers made a good case of how our microbiomes have changed because of human activities and also because of the way these systems have evolved, especially over the last century. And one of the things that we have to consider when we think of future of health vis-a-vis microbiomes is the effect of climate change. So that's where I would like to begin by focusing on what might be some of the impacts of climate change on our health. Good, thank you. So it's projected, as you know, that we will have a few degrees centigrade increase in temperature on our planet because of the rising levels of CO2. And the estimate is by 2100, we will have a 4-degree rise in temperature. And by 2030, when we will have about half to 1 degree rise in temperature, we will already begin to see some of the devastating consequences of this slight increase in what appears as a slight increase in temperature. And it's projected that there will be 250,000 deaths that could be attributed to infectious diseases that will spread in ways that the increased temperature supports in regions where they haven't been previously. And there are about 17 different tropical diseases that are projected to spread in places that they never occurred before. And it's estimated that about $2 billion will be spent, in fact, we're doing it right now, per year to prevent or fight some of these diseases. So that's just considering an increase in temperature. If we take into account issues of antimicrobial resistance, if that goes unchecked, the situation could be far worse. And there it's projected that you can have up to 10 million deaths that could be attributed to infectious diseases if antimicrobial resistance continues to rise at the current pace. And so there's some big questions about how we're going to fight this increase in resistance. We have to go down to basics in understanding how resistance evolves and how we can be much more rational in our approach to fight resistance and also to combine drugs to prevent resistance from occurring in the first place. In addition to the issue of resistance, there's this issue of how microbiomes have changed because of anthropogenic activities, such as microbiomes in our agricultural sector. And the previous speaker has shown how we've modified our own microbiomes by taking antibiotics. But if you look at the microbiome at large across the planet, and especially on arable land, by using nitrogen fertilizers and pesticides and so forth, we have completely modified plant microbiomes. Plants have not co-evolved with their microbiomes in a way that you would naturally see. And as a result, the plants tend to be a lot more dependent on our intervention so we can have the high productivity that is necessary to feed the population. And there are estimates of how these interactions could be affected. And these are microbial interactions, trophic interactions that happen in the oceans and also in soil. And the microbes are small, but they account for 10 to the 30th different cells on this planet. And they are highly processive. So they contribute to nearly 1,000 times more productivity than some of the macro species on the planet. So when you shift the climate, or when you use harsh means for agriculture and shift microbiomes, you run the risk of diverting some of these or shifting some of these cycles. Just to give you a sense, in the oceans, microbes are implicated to be involved in processing up to 60 gigatons of carbon every year. And in the soil, it is said if respiration were to stop, if microbial respiration were to stop, it will take just 12 years for the photosynthesis to consume all of the carbon dioxide in our atmosphere. So these are some very, very important systems, and we understand very little about how to quantify their resilience and how to understand how to manipulate these organisms when necessary in a responsible way so we can prevent things like resistance and tolerance and so on. So microbial ecology has a very central role to address these issues. On the one hand, for ecosystem management, we need to be able to predict when systems might collapse and when they might lose resilience, or when their behaviors may shift to states that are less desirable for cycling nutrients. And on the side of infectious disease and global health, we would like rational means to understand how organisms evolve tolerance and how we can prevent tolerance from arising, and how we can use combination therapy to fight resistance. And all of these capabilities will have underpinnings in some very basic research into cellular responses, interspecies interactions, and quantifying some of the emergent phenotypes in a systems framework. So I'll tell you two stories. The first story will be about trophic interactions between two groups of organisms and how that evolves over time and how resilience in that system can be quantified. And the second story will be about drug resistance in TB. So I'll speak more about tolerance and resistance as to how Mycobacterium tuberculosis sees the world around it and triggers reversible responses to fight the drug that we're using to try to kill it. So these are the two major topics that I will be speaking to you about. And the implications of this are both in terms of ecosystem management and also in global health. So when you think about microbial communities, and the interaction I will specifically speak about is an interaction that happens between two groups of organisms that live a few centimeters below the surface in soil. You find them in sediments and ruminants and so on. So they're widespread. And these are a group of organisms called sulfate-reducing bacteria that break down complex organic compound using sulfate as the electron acceptor. But when sulfate is depleted in the environment, it starts interacting with a group of organisms, the archaea. And so this is an interaction across the two domains of life. And this interaction then drives the processing of several gigatons of carbon to methane. And as you know, methane is a greenhouse gas, and that gets ultimately released. So this cycle here plays a really important role in processing in large amounts of carbon, and there are some numbers given here, through the soil system. It's not clearly understood how these interactions evolve. And this is a big challenge in microbial ecology, is trying to track at a molecular and systems level the interactions across different species to be able to then predict their behaviors in changing environments. And so we often use this concept of synthetic communities. And these are communities that are established in the laboratory to simulate some of the interactions you observe in the natural environment. And this particular syntrophic community was established by David Stahl at the University of Washington, along with John Lay, who's also at the university. And this is a really interesting system for several reasons. One, it does simulate a natural trophic interaction, as I described earlier, but it's between two organisms that have never seen each other before. So Desulfovibrio and Methanococcus and the specific species that they selected had never seen each other before, but they have physiological capabilities that are complementary in a way that makes ecological sense. So Dave Stahl and John Lay put these organisms together in a situation where they are obligately dependent on each other. So in normal circumstances, DV or Desulfovibrio can break down organic compounds like lactate using sulfate as an electron acceptor. And in the process, it produces energy. But it starts producing hydrogen and all these other intermediates. And when they build up, that can be inhibitory to the reaction. So if sulfate is missing in this environment, Desulfovibrio cannot grow. However, if this methanogen, Methanococcus maripaludis, is present, then it takes advantage of this organism because it can utilize the hydrogen and the formate and acetate and break it down further into methane. And this becomes now a favorable reaction. So the two organisms now depend on each other and they start living together. So they set this community up and set 24 different lines and allowed them to evolve in the laboratory to see how the growth characteristics might change over time when they're forced to interact with each other. And what they observed was remarkable. Within 300 generations, this synthetic immunity improved dramatically in terms of its growth rates and also the yield. So they were growing 80% faster and producing 30% more biomass. And this was observed across all 24 lines, which is quite remarkable given the short time frame in which they have learned to take advantage of each other. So this really lends itself to a deep systems analysis of why this happened. And the first step, because of the technologies we have now, we can go and sequence the genomes of these organisms, and we did that across all lines, and found that mutations that accumulated at a unprecedented rate. We had never seen mutation rates to be this high. And there might be a mutator phenotype that is introduced because of the stress. But regardless, there were hundreds and there were thousands of mutations across the genomes of both organisms. And the most striking among them was that the genes that are involved in the pathway for sulfate respiration in desulfovibrio had accumulated loss of function mutations. So that pathway was eroded across that group of organisms. And that, in retrospect, makes sense because sulfate respiration is no longer going on. So it was a dispensable function. As a result, mutations that were loss of function were allowed to accumulate, but that it was selected was quite interesting. And this is a hallmark capability of this organism. A second observation caught our eye, and it's something many people have reported previously. The regulatory networks of both organisms had accumulated mutations. Now, gene regulation is a capability all of us have, microbes have, and the intent of that capability is to turn on and off the expression of genes and processes as required. And so in this case, it seemed initially surprising, and it's been reported that when you have adaptive evolution, some of the first systems to get mutated are the regulatory systems. Why? It's never been clear. People have put forth many different hypotheses, and so did we. We said, since we have an obligate relationship between these two organisms in a uniform environment, it doesn't really make sense to have regulatory systems because you don't need to turn genes on and off as much, and that is the reason why this is getting mutated. So that led us to the hypothesis that that regulation is unnecessary in an environment that is uniform. So the corollary to that would be that if the environment is fluctuating, then regulation would be really important. And this was a hypothesis we could test. And this has a lot of implication in understanding evolution of different phenotypes, including things like antimicrobial resistance. So in this case, we said, okay, we can set up a system where we take the sulfate-reducing bacterium and we force it to live in a fluctuating environment. On the one hand, we give it sulfate, so it can grow independently, respiring sulfate, breaking down lactate. But then we will shift it to an environment when there's no sulfate. So it will then grow with its counterpart, the methanogen, using an interaction called syntrophy. And so if we make this organism go back and forth between sulfate respiration and syntrophy, that fluctuation over time will make sure that the regulatory systems that are required for turning on the capabilities for each of these two lifestyles will be intact and refined. So when we set this experiment up, we had the most unexpected result. Within 3 to 7 transfers, the wild-type culture collapsed and went extinct. And we did this many times, and like clockwork, within 3 to 7 transitions, the wild-type cultures collapsed. By contrast, a single mutation in the regulatory network rescued this organism, this community. And the single mutation was in the system for regulating sulfate respiration, which was very counterintuitive because sulfate respiration is something that needs to be turned on and off when it's going between the two environments. So it did not make sense why it would do so. So we then went on this discovery process to figure out what had happened. And when we first looked into the transcriptome changes across the different phases of the experiment, growth in syntrophy or growth in sulfate respiration or growth across the transitions, we saw that over twice as many genes were being regulated in the wild type as in the mutant cultures, and which was, again, supporting the view that the regulatory system is required to turn genes on and off for sulfate respiration. So we built a model to understand what are the consequences of having regulation when the environment fluctuates. And the model made a prediction that over time, as the environment fluctuates, essential components within the cell are going to get diluted out. And the thinking behind this was the following. When the organism is growing in a non-permissive condition, it is shutting down enzymes required for that condition. And then when the organism divides, the concentration of those components within the cell gets diluted out. And then we shift it to the environment where they need to be made again. And so it starts making it again, which is an energetically expensive proposition. But if it was a single shift, it would make sense to make that expenditure of energy. But then we shift it back to the non-permissive condition. And as a result, it can never fully restore all the components that it is losing in the non-permissive condition. It's a great hypothesis. We had to go and test it, so we did whole transcriptome measurements, and sure enough, in the wild type, essential transcripts get diluted out. You look at proteins, they get diluted out, and this also appears at the physiological level, where hydrogen production and lactate consumption start going down. But in the mutant, it reaches a steady state. So this really gets at a very fundamental property of regulatory systems. It's a burden to have regulatory systems, but it makes sense to have them when the environment changes seldomly, because you can be very adept at making things only when they're required. But if the environment becomes unpredictable, at that point, it doesn't make sense to constantly turn genes on and off. And that becomes energetically burdensome, as I'll show you in a minute. And when we looked at single cell level, it told us why these communities collapse. So these are single cells, and we took every cell, and within that cell, we interrogated the abundance of about 90 essential transcripts. And what we saw was there was a lot more cells in the wild type that had abundant amounts of these essential transcripts as compared to the mutant. There was slight difference, not too much, but within about 3 transfers, so we went sulfate respiration to syntrophy, to sulfate respiration to syntrophy and back, most cells in the wild type no longer had the essential transcripts. And as a result, they were not adept at acclimating to the new environment. And around that period, we start seeing that cultures across different replicates start collapsing one after the other. And this is a foundational insight, because it tells you when our environment is going to change unpredictably. And it is going to happen with extreme drought and unpredictable rainfalls and events that we are beginning to see that are unprecedented from the evolutionary standpoint of what these organisms have experienced. These unpredicted changes can have drastic consequences on the resilience of these systems. And we can look at the overall internal state of these systems to see whether there are signatures that can predict collapse. And this could also be used for ecosystem management, so we can track systems in the wild. What we see in the wild type is that as we go through transitions from syntrophy, sulfate respiration, syntrophies, it starts moving away from the preferred state that is required to live in those conditions. And it looks very different and ultimately reaches a point that is so unstable that the cultures collapse. But a single mutation can rescue it, and that shows you the power of these microbial systems, that they can adapt very quickly. Although we have simulated this in the laboratory where they're growing really fast, they may not have that privilege in the natural environment where they don't divide as quickly. So evolution takes much longer in the environment than it does in our laboratories. But a single mutation now allows the organism to create an internal state that is appropriate for the environment in which it lives. And it can do that ad infinitum. This goes back and back and back. And so we can look at the cost of doing this, and we see that in the wild type is expending a lot more energy to adapt to the new environment, because it has to constantly try and make the components that have been diluted out. And this energetic cost becomes unsustainable, eventually leading to collapse of the organism. So this work was, the first part that I shared with you, was done by Sardar and Anne with a team of collaborators at ISP and at other institutions. And it showed us quite a few things about the properties of microbial systems, that frequent environmental fluctuations can lead microbial populations towards collapse. And the reason for that is because they are over-regulating their genome, and as a result, they are diluting out essential components, but a single mutation can rescue this. this framework, we are now developing it into a stress testing framework to quantify microbial resilience. And that is a very difficult property to quantify in the natural environment, because you have to make measurements longitudinally over a really long time frame. What we are setting up now is akin to a cardiac stress test, where we can take the microbes, put them in a reactor, which will be like a treadmill, and make them run, which is make them do their different physiological tasks, but constantly change the environment, so they have to keep changing exactly what processes are turned on and off. And as a result, we're pushing the organisms to an extreme to see their response and recovery times as a function of stress and a function of transitions. And the time when they collapse will be very characteristic of their resilience. So the systems that are more resilient will be able to sustain these transitions over a longer time frame, whereas those that are unstable and about to collapse will succumb to transitions very quickly. And this type of a system could then be taken off-site to the locations where we are concerned about environmental changes and the impact on the field community, so we can set up monitoring systems as to how those communities are doing. Now, the flip side of the story is that of drug tolerance. On the one hand, you see that when the environment fluctuates too quickly, you can have systems collapsing out. Now, the other problem we have is we want to kill systems that are causing diseases, and they are able to overcome everything we're throwing at them, and that is tolerance and resistance of infectious agents like TB. And it's estimated that a third of the world's population is infected with TB, and there's about 1.5 million annual deaths that can be attributed to tuberculosis. And the big problem in the field now is the emergence of resistant strains. So you had the multi-drug resistant strains, the extremely drug resistant strains, and now you have the completely drug resistant strains that cannot be killed by any of the antibiotics we have. So there's an urgent need to figure out how to overcome resistance and tolerance, and to come up with new drugs that can be used to fight the spread of TB. And over the last 40 years, there's only been one drug that's been approved to treat TB, a new drug that's called bedaquiline. And when bedaquiline was used, they saw that there was a delayed killing effect, that over the first week to 10 days, there was no killing. And we asked, can we use this as an example to understand how TB is able to tolerate this drug and come up with a rational approach to fight such tolerance mechanisms? Our hypothesis was that the microbe is able to elicit a tolerant phenotype when it sees the drug, and this phenotype is generated by its regulatory systems. So if we can understand what those networks look like, then we can use drugs that can render those networks insufficient or disrupt them, so we can then have the drug be more effective. But the challenge with TB is it's hard to grow in the laboratory, and there aren't as many laboratories as you have for systems like yeast and E. coli that have generated wealth of information that people have used to generate networks of these organisms. So we had an organism that doesn't grow well, is not as well studied, and we had come up with a hypothesis that required us to know the complete network inside the cell. So to do that, we came up with this data-driven approach that we published several years ago. And the idea here is to take an organism, in this case TB, and expose it to different kinds of environments it might experience in its natural life cycle: low pH, low oxygen, nitrosative stress. And we did this in collaboration with the Center for Infectious Disease Research, with David Sherman and also with Alan Adurham, who's going to be speaking tomorrow. And we generated a large compendium of transcriptome profiles. So this is a view into how TB sees the world around it. And we took all of these data, and then we applied statistical learning and machine learning algorithms to these large compendia of data. And along with the expression data, we used the genome sequence, protein-DNA interactions, protein-protein interactions, evolutionary associations, and so forth, to then reverse engineer the network inside TB that must have been responsible for making all those, sensing all the changes we imposed on it, and for eliciting the responses that we measured across the different conditions. So the model is a network model that is predictive, and we've shown that we can take a model like this and take completely new environmental conditions, take a few measurements and predict how the organism is going to respond to the new environmental condition. So now that we had a network like this for TB, we asked, okay, now if we expose it to bedaquiline, this drug that FDA has just released, can we figure out how it is able to tolerate this drug treatment? So we can then drug that network. So we took all the expression changes for TB and we analyzed it in context of the network and saw that it was triggering a few subnetworks, 2 subnetworks in particular, that were allowing it to resist treatment with bedaquiline. And we zoomed into this network and we saw that there were roughly 70 genes in one network and roughly 30 genes in the second network that were associated with various cellular processes like virulence, detoxification, and adaptation, changing cell wall structure, and so forth, that were collectively responsible for helping TB overcome the drug treatment by bedaquiline. It's very difficult to target all the genes if you want to overcome tolerance. But because we know the structure of the network, we can focus specifically on the hubs of this network and ask, if we were to remove those, would we make TB more susceptible to this drug? Okay, so we also were able to figure out why TB was able to elicit such a specific response to a drug that it had not evolved with. So what's the mechanism? Bedaquiline inhibits ATP synthase, and that's the cause of death. What the network told us was that by inhibiting ATP synthase, it was creating a state that looked very similar to the nutritional stress state. And this is what TB is sensing. It is not necessarily recognizing this molecule. It's recognizing the consequence of the action of the molecule. And that triggers this network, which is able to then upregulate processes that inhibits bedaquiline effect on TB. So our hypothesis was, if we knock these 2 hubs, we should be able to make TB more susceptible to bedaquiline. And we showed that to be the case. So we removed each of these regulators from the genome, and voila, it was more susceptible. So now the next thing we had to do was figure out if there was a small molecule that could accomplish the same thing. And we scoured through all the drug response data sets that we had and identified a molecule called pretomanid, which was targeting one of these 2 regulators called 880, and we predicted this regulator will make this tolerance network partially dysfunctional and make TB more susceptible. And we showed that to be the case, and that so when you added pretomanid and bedaquiline together, it made TB a lot more susceptible to the 2 drugs together than the individual drug. And we showed that this mechanism of combinatorial action was through this particular network because we could overcome the effect of the combination therapy by overexpressing that regulator. So this study was published just last year, and it showed that we can rationally use a systems network-based approach to find drug combinations to fight drug tolerance in a system like tuberculosis. And in summary, there's 2 sides to this story. One is the resilience of microbes and how that is a crucial property that we need to be able to quantify and understand in consequence of systems responding to climate change with regard to food security. And on the other hand, we can use the same approach to understand drug tolerance and come up with rational approaches to fight resistance in microbes. And what it tells us that the regulatory networks within these organisms are very facile in being able to evolve adaptively to environmental change and restore resilience with few mutations, but that also gives us capability to understand how we can trick these organisms with combination therapy to overcome their tolerance. So with that, I'll just thank the group that was responsible, especially Sardar, who did the first part of the work, and also Eliza, who was the person who spearheaded the tuberculosis work. So I can take a couple of questions before lunch. John.
Hi. First study was really interesting, Nitin, but the question I have about it is with respect to bet hedging. So one would have expected there would be a small population of cells that would be able to predict a new environment. even though the environment was not one they've seen before. And this is the principle of bed hedging, and I wonder, even when you looked at the single cells, you didn't see examples of bed hedging, or did you?
There was actually, the way the data was presented in the slide wasn't showing that behavior. So bed hedging would be when you have a small subpopulation within the larger population that is doing something slightly different in anticipation of some environmental change that would allow that subpopulation to render the overall population stable. And in this case, when you plot the data in a multidimensional space, in a two-dimensional space, you do see subpopulations emerge. And so when the cells are growing in one condition, you see that there's a subpopulation that is in a state that appears to be more appropriate for the other condition. So when you shift, that subpopulation takes over, and as a result, you have this adaptive behavior. And what we see is just before collapse, that subpopulation disappears. So that also supports some of the view that the robustness of these systems comes from their capability to generate these large numbers of subpopulations. Same is true of tolerance as well, is that you would have TB, for example, generate many subpopulations, each subpopulation with tolerance or resistance phenotypes for different drugs. And depending on an unexpected attack, you would always have the overall population be ready so it can overcome those attacks.
Okay, Nitin, thanks for a wonderful talk. So I have a question about the first part. You talked about how when you cycle the environment, the community would collapse. So does it depend on the period? I mean, the frequency of cycling seems that if you make it slow cycling, maybe they have time to adapt. And I have also another question about the second part of your talk, which is that...
Go ahead.
Okay, so maybe you can answer the first question.
Yeah, so the first, you're absolutely right. I think the frequency and period both matter. So the period in any given condition, if it's long enough, then you could have the organism restore all of its function. And then it would justify the need for control. So there's a trade-off between the amount of energy expended in regulation versus amount of energy or amount of fitness gained because of the regulation. So right now it is it's spending a lot more energy than the fitness gained, so it is collapsing.
Right. And then with regard to the second part of your talk is that when you do these environmental perturbations and look at the phenotypes, generally, how would you tell you have done enough of these experiments to constrain the model such that it would give you the predictive power that you want?
See that this is the second part of the...
Okay, so second, my understanding is that for any organism, you developed a very powerful method, which is that you subject the organism to different environmental perturbations and look presumably at how transcript profiles correlate with each other and how they correlate or anti-correlate. But then my question is that at which point would you say, I have done enough of these perturbation experiments that I understand the network enough now that I could now...
I see.
Yeah, I'll now make useful predictions.
So how many experiments would you need to
become
predictive?
Right.
And it's a tough question to answer, but I think it's the type of experiments that you do that, and the diversity of conditions you choose that makes that assessment. Because if you use the same few environmental conditions and you just repeat the experiments, you don't cover the space of possible phenotypes the organism is able to generate. But and that's the reason we typically use two dimensions. One is the environmental dimension, where we pick many different environmental conditions that are ecologically and evolutionarily meaningful for that organism. Then we also use the temporal dimension. So for every perturbation, you have to look not just at the dosage of the perturbation, but the temporal response, because you need to capture the sequence of event, including causality.
Thank you.